Hi, and welcome to the Hood Bioinformatics program. I'm Miranda Darby, your advisor and the director of the program. I also am the advisor for the Hood Bioinformatics Club and other activities. I'm happy to work with you as you progress along the program, either to help craft the correct progression of courses that fits your needs the best, or to help you with things like internships or job applications. You can always contact me at Darby via email at darby@hood.edu, which is always the best way to get a hold of me. And we can also arrange either Zoom meetings or in-person meetings to fit your needs. I really look forward to working with you and watching how you grow and progress as you complete the program. Thank you. I'm going to tell you a bit about what it's like to go through the program, what we hope you'll learn, and how to progress. So first I'll be talking about our program's learning outcomes, which are the things that we hope you'll learn over the course of the program, and also how our program aligns with the learning outcomes that are suggested by the graduate school. And then also, I'll be presenting a couple of suggested course progressions for full-time or part-time courses of study. And I'll be telling you a bit about the unique opportunities that are provided by the bioinformatics program, which hopefully will help your career. So over the course of the program, the things that we hope that you'll be learning include programming languages, including R, Unix, Linux, and Python, since these are the three most important programming languages that are most frequently on job ads for bioinformatics positions. In addition, you'll be learning about algorithm development and testing, machine learning, data management, database systems, and SQL. And the use of publicly available databases and analysis tools to perform biological research. The types of research that you'll be doing include proteomics, genomics, and metagenomics, so the study of proteins, the study of genomes or genes, and the study of the microbiome or other collections of microbes. In addition, you'll be working to learn bioinformatics sequence analysis, so how to analyze sequences and compare them to each other and compare whole genomes to each other. In addition, you'll be learning some structural bioinformatics, how to model the structures of proteins and RNAs, and how to model expression of genes and model those into biological systems. So how some genes might go up, some genes might go down, how do those all fit together? And in addition, we'll have a focus on leadership and project management. And really the focus will be on leading and performing reproducible research. The graduate school's core outcomes include the ability to communicate effectively, the ability to perform research, critical reasoning, problem solving, ability to think in a diverse way and recognize diverse ideas, and to act in a professional manner. And all of these things will help you as you progress in your careers and in academia. So the course progression, the entire program is comprised of 11 courses, or 33 credits. Although some students are better off taking an additional course, taking both our foundational courses. So I have listed here a suggested progression of 2 courses per semester, although someone who wants to take 3 courses on some semesters certainly can. So you can take 1, 2, or 3 courses every semester, and you can progress at your own speed. As long as you complete the entire program within 7 years, you can kind of go at your own pace. And I will certainly help you to do so. So again, to contact me as your advisor, I recommend email, and my email address is darby@hood.edu. So our two foundational courses are BioFX 501, which is our foundation in biology, and BioFX 502, which is our foundation in computer science. So students who have not recently taken an upper level, 300 level or above genetics course or cell biology course are strongly advised to take BioFX 501, and in some cases required to take BioFX 501. Students who haven't had formal training in computer science are required to take BioFX 502. And it's recommended that you complete both of these courses as early as possible in the beginning. At the same time, you can take BioFX 503, which is our biostatistics course, which is performed in R, but you don't need to have any R programming skills in order to take the course. You'll learn everything you need to know along the way. In semester 2, I recommend taking Leading Reproducible Research, which is BioFX 545, which will focus on good scientific design, study design, scientific method, and also how to lead reproducible research when you're working in large matrix environments that include biologists, bioinformaticians, computer scientists, and biostatisticians, who all think about things in slightly different ways. And so this is a way to kind of wrap our minds around how to communicate with different people with different ways of doing things. In addition, you'll be learning about bioethics and good responsible leadership. At the same time, I recommend taking BioFX 551, which is programming for bioinformatics. So our initial programming course is in Python, and BioFX 551 will introduce you to R and a little bit of Unix Linux in order to get you going in those other languages. But semester 3, I recommend taking BioFX 552, which is Bioinformatics Applications 1. Bioinformatics Applications 1 should be renamed Bioinformatics Data Skills. It's all about the nuts and bolts of how to access online data resources, to use a remote compute cluster, which is something I'll be talking about a bit later, and other kind of nuts and bolts skills for basic genetics and genomic studies. In addition, I recommend taking BioFX 530, which is our database course, which will teach you a bit about databases and SQL. The following semester, I recommend taking BioFX 553, which is an applied bioinformatics course, which includes applications of the skills that you've learned in BioFX 552, but also how to do basic types of analyses, gene expression analyses, some machine learning, and so forth. And at the same time, I recommend BioFX 550, which is our course on sequence analysis. So BioFX 550 is our course that it's sort of what people think of as classic bioinformatics, how to effectively use BLAST and NCBI resources and so forth to find and analyze genes and proteins. The following semester, I would recommend BioFX 548, which is our data visualization course, which will help you to effectively plot and present your data in lots of different kinds of studies. And BioFX 572, which is an applied bioinformatics course, which kind of serves as a practicum for our certificate course, but is also available to you as an elective, and that's a hands-on course where you'll be creating scripts that you can reuse, that you can perform gene expression analyses, metagenomic analyses, and other types of studies. And then finally, most students conclude with BioFX 590, which is our capstone course. And I'll be talking a little bit more about capstones and theses and internships and things that you can do. But every student performs some sort of hands-on research and actual first first research, original research, in order to complete the program. So if you're a full-time student, and this includes the HRSA scholars and international students on an F1 visa, they're required to have 3 courses per semester. And of course, any other student is welcome to take 3 courses per semester if they wish to. So for the full-time progression, what I suggest is starting out with EAP 500 if that's needed, and that's something that will be communicated to you by the grad school office. So that's dependent on TOEFL scores, etc. Any student who completed already a degree in the United States doesn't need to take EAP 500. If you don't need to take EAP 500, I recommend taking BioFX 501 and or BioFX 502. And again, if you're required to take both of those courses, that should be communicated to you at the time of your acceptance. But BioFX 501, again, is our foundation in biology, and BioFX 502 is our foundation in computer science. And if you have an additional slot, if you don't need to take all 3 of those, I recommend taking BioFX 503, which is our introduction to biostatistics. The following semester, I recommend taking BioFX 545, which is leading reproducible research, BioFX 550, which is sequence analysis and protein modeling, and BioFX 551, which is programming for bioinformatics, where again, you'll be learning about R and other programs, or other programming languages. The following semester, I would recommend taking BioFX 552, which is our data skills applied bioinformatics course, and BioFX 548, which is our data visualization course, and BioFX 530, which is databases and SQL. And the final semester, I would recommend taking BioFX 553, which is Bioinformatics Applications 2, which includes applied bioinformatics and some initial studies into gene expression analysis and so forth, and also includes some machine learning. And BioFX 546, which is a whole class on logic and knowledge and machine learning, and 590, which is the capstone course. So there are several unique opportunities that will be available to you throughout the course. The first is the BioFX Slack channel. I maintain an active BioFX Slack channel, and most classes also have a channel on the Slack, in a sub-channel within our Slack. Slack is frequently used in the computing industry and in bioinformatics labs for people to easily collaborate across labs. It's sort of a chat messaging service, and yet is very much designed for business situations. So you can easily set it so you never get notifications at night, it's non-intrusive, and no one gets to know any of your personal information. So this lets you privately contact all of the other students in the program to collaborate on projects, or just to chat, or to work together on homework, or so forth, without having to share your personal information or phone numbers with each other. In addition, there's the Bioinformatics Club, which I'm hoping will become more active in following years. I'm hoping to do things like journal clubs, fund drives, maybe a hackathon or two, but it's a student-run club, and so it will depend on your energy and participation and willingness to make it a great collaborative space. In addition, you'll be using in many of your courses the BioFX computational server, which is a Slurm server. And I've very carefully designed this server to mimic the way that most people work on large big data projects, such as gene expression analyses, next generation sequencing, and so forth, which often require way more data than can fit on your computer. For instance, my last big next generation sequencing project used 6 terabytes of raw data just to start out with. And so in order to effectively use and process all of that data, one needs to work on a remote supercomputer. And it requires some kind of acquired skills that you'll be learning along the way. Unfortunately, it's very possible to accidentally delete a lot of data and other things. So we've created a sandbox for you, this sort of training wheel server, where you're not going to do a huge amount of damage to anyone, and you can learn to become competent and confident so that when you go on to the NIH or other places to do work or internships or capstones, etc., you'll be comfortable. So we've purposefully designed our server to mimic the one that's used at NCI, the National Cancer Institute, since many of our students end up doing internships there. That brings me to our practical hands-on research opportunities. In a typical year, there are many internship opportunities available in the Frederick area, both at local biotechs and at the National Institute of Health, both in Frederick and in Bethesda. There's also some spots in Shady Grove and in Gaithersburg and so on. So I frequently post information about capstone or about internships and job opportunities to the Slack channel. So you should immediately sign up for Slack and start applying to those opportunities. I will be sending out an email to everyone shortly after the start of term with information for how to join the Slack channel. As I mentioned, the program culminates in a hands-on capstone. And there are a couple of different ways you can complete the capstone requirement. One is to complete an internship and do hands-on research in an outside laboratory. Either during the summer or during one of the semesters. And what you need to do is contact me beforehand and I will help you set it up as part of your capstone. I will help collaborate with your advisor and help set what the project will be, what will be done, what the time frame will be, and so forth. And this will help keep everyone on the same page and make sure that you don't end up doing way more work than you need to, but at the same time that what gets done is done well and everyone is satisfied. Alternatively, I offer a capstone course in the evening. So those of you who are either not able to find an internship or don't have full-time employment and don't have time to do a 9-to-5 style internship elsewhere can do research in my lab. So I have a number of ongoing research projects and I will invite you to participate in them and I will give you some suggested projects. And together we'll work through some original research that is hopefully even going to be written up. So I'm actually working to write up one of last year's capstone, actually 2 of last year's capstone projects as papers with the students who completed them. An alternative way to complete the program is through a thesis option. And so to do a thesis, instead of the capstone, which is 1 semester, the thesis is 2 semesters. And so you should tell me actually in your first year if you think you're planning on completing the thesis option, and I can help you arrange with a laboratory that will be your host laboratory for the thesis. There's one other major difference between the capstone and the thesis. So the capstone is a proposed project that's expected to take 180 hours of work. And you need to keep track of your hours and how you spend it. But we all know that science doesn't always happen the way we think it's going to happen. So the capstone gives you a designated framework where you're going to work towards these goals, but as long as you're continually progressing towards the goals, if things happen and you're not able to complete everything that you initially had outlined, that you had planned to complete, that's okay. As long as your mentor and myself agree that you're making good progress, then there's no problem and you'll be completed once you've completed the 180 hours of work. For the thesis, that's more self-driven research, and it's your own sort of idea. And so there you need to actually complete your work that you've proposed in order to complete the thesis. So theses can be quite a bit more time-consuming than capstones. And I'm happy to talk to you about both of these options in the future. And again, you can email me at darby@hood.edu to discuss any of these options. So welcome to Hood College. I'm excited to have you here, and I look forward to meeting you in person.
Hello, my name is Bianca Hoke, and I am a graduate student in the Hood College Bioinformatics program. I would like to extend an official Hood hello from me to you, and a congratulations on your acceptance. You are more than ready to start this new chapter in your life. The Bioinformatics program is full of brilliant minds, and I am sure you will learn a lot during your enrollment. Whether you want to learn more about computational genomics, proteomics, or automating your analyses using Python or R, this program will give you the resources and tools you need to jumpstart your career as a bioinformatician. So, be good, be Hood, and carpe diem. Take care.